Okay, so I'll start the meeting in about a couple of minutes, right? So in case any of your colleagues are joining, we'll let them join so that we are not interrupted during our class. So we'll give them a couple of more minutes and then we'll start with our regular session. Okay. Mm, all right. I hope I'm visible and audible to everybody. And in case there's an issue with the you viewing my slides or screen whenever I'm sharing it, please prompt at that very moment so that I'm not just blabbering. And that is just it. And we'll be starting. So what I'll be dealing with today is basically the fourth week's assignment for the past year, which might help you with your final exam for this particular course. Along with that, we are going to deal with some concepts that were dealt with in week four. And since there were just two classes in week four, there were not many conceptual queries also that I came across on the discussion forum. So, along with that, what I've done is I've added some these practice problems for restriction mapping. So that is the part we are going to do at last. We'll start with the previous year's questions today and we'll end with restriction mapping problems. I also wanted to add one or two haplotype related these numerical problems. They are pretty huge numericals. If I would have added one, it would have taken at least half an hour to 45 minutes to discuss. So for now, I'm not added any. Probably, if at all we get to meet again, I'll deal with that, right? So a few people were joining. I hope everybody has joined by now. Okay, I'll be sharing my screen. If at all there's any problem with that, let me know, right? So just a moment. Okay. Somebody in the lobby. All right. Okay, so over here, the first thing that we are going to do is discuss the previous year's assignment for week four, as mentioned in the slide, right? So moving on, we'll just deal with questions. I'll just do it as I normally do. I'll read the question. I'll give you a moment to think about or ponder upon what could be the answer, and then we'll actually discuss the answer, right? So I would have loved it if it could be an interactive session, but there are certain limitations software wise, right? So this is how I have been dealing with it. Okay, so question one. Okay, I am on the first slide. I hope everybody can see it. In case there is an issue, please let me know. Okay, so your question one is, which one of the following statements lists the goals of the HapMap project? Okay, so in order to solve this question, one thing you should be aware about is the HapMap project. So the options given are to determine the common patterns of DNA sequence variation in human genome, to identify common variants likely important in the risk of common disease, or to make genome related information freely available in the public domain, or D, that is all of these. So it is a very theoretical questions. Most of the questions in this particular week's assignments were theoretical. So I don't think they'll take much time to solve, right? We can directly, people are joining. I'll just have to go back to the lobby for a moment. Okay. Okay, so for the ones who have actually read the question, I did not mean to give this much of a gap, but there were people joining. For the people who have just joined, maybe you can read the question once. I'll not go through the options. Maybe you can read them. The question is, which one of the following statements lists the goal of the HapMap project? So these are the particular options that are given. We have gone through them once, and now we want to see the solution, right? So the solution for this particular question is all of these, right? Just a moment. Yeah, so International HapMap project, it was basically launched in October of 2002, right? It was after the first rough draft of Human Genome Project had actually been published in Science and Nature, right? After that, this International HapMap project was launched, and what it aimed at was to identify common genetic variants among people, right? Also, along with this, it like why would anybody need to identify common genetic variants, right? It was basically because some of these genetic variants which normally exist in a population, they could be risk factors for certain diseases, right? So diseases, when we talk about these common diseases, they are not really monogenic diseases. That is, they do not originate from a single gene, right? Multiple genes or multiple alleles at different loci are involved in contribution or manifestation of this disease. And sometimes having one particular genetic variant could basically make you susceptible to get a disease maybe in future, right? Okay, I'm just viewing the lobby. There were some more people, I had to admit. I'm so sorry for these interruptions, but your colleagues also, uh, the ones who are joining late, I have to admit them, right? So, okay, so what was the purpose of this HapMap project? It was to basically assess normally present variations which could act as risk factors for particular diseases, right? And the third statement is also true. That is that just like the human genome project that was made public, right? After it was done, yeah, after it was completed, the sequence information was made public. Similarly, this sequence was also uh, aimed to be made public so that it could be used both in the research as well as the clinical domains, right? So moving on to the next question. Okay. In a polygenic disorder, the term genetic risk factor refers to. So genetic risk factor is something that we were just discussing with respect to what HapMap project intended to do. So it intended to basically recognize the genetic risk factors among other things, right? So over here, there's this question which is basically focusing on the term genetic risk factor. What does it mean? A disease associated gene variant inherited from the parents. B, a gene variant that confers a small yet significant risk of developing the disorder. C is a gene variant that is detected in the normal population. And D is all of these. So anybody who has just followed what I have said would have a pretty good understanding of what might be the answer, right? So we'll not wait over here for a very long time. I still think there's somebody who needs to be admitted. I'm so sorry. I'll just go back for a while and I'm sorry to give that answer also. Okay. So your friends are joining pretty late today. Okay. For now, I have admitted people. Again, I hope my screen will remain visible. Okay. Yeah, I've checked. It's visible. So the answer to this was basically, again, all of these. What are polygenic disorders? Polygenic disorders are basically disorders that involve multiple genes which contribute to that particular disorder or disease, right? Okay, so why is the answer over here all of these? Because what is a genetic risk factor? It is basically a kind of genetic variant which is, first of all, it is present in the normal population, right? It will, what does the normal population over here means? Means somebody who might not be affected by that particular disease might also have this genetic variation, right? So it is present in the normal population. Apart from this, it confers a small yet significant risk of developing the disorder. So, since it is present in the normal population, not all individuals that have this genetic variation would basically get the disorder, but it would put these individuals at the risk of getting this disorder, right? So, this statement is also true. Also, it could or could not be a disease-associated gene variant which is inherited from the parent. How could this statement be true? If, for example, this particular variation or genetic risk factor occurs as a polymorphism, right? So that polymorphism might have occurred in your parent also. And when that parent will, when you were conceived, basically you got that particular polymorphism that is a genetic risk factor for, say, diabetes. So you might have inherited it from your parents as well, right? Moving on to question number 3 now. Okay. Ha. Let me just get the laser. Ha. Which one of the following statements distinguishes a monogenic disorder from a polygenic disorder? So again, this is a very generic question, right? Let us see its options. DNA sequence variation contributes to the disease onset, can affect either sex, or the affected individual can transmit the disease causing allele to the next generation, or the fourth option, which is none of these, right? So I think I don't need to spend time or give you time to assess this question, right? It was pretty straightforward. So we'll just come on to the discussion part. So over here it says, just a moment. Yeah. So over here, the correct answer is none of these. Why? Because what is the major difference between a polygenic and a monogenic disorder? So for that, I have included this slide and I have been saying it for the past couple of questions also. What is a monogenic disorder? It is basically one in which variance is present in one particular gene only, right? On the contrary, what is polygenic disorder? It is basically a disorder in which variance in multiple genes is present at the same time, right? So there are certain examples listed for monogenic and polygenic disorders. Monogenic disorders, a lot of examples we've also studied when we were studying pedigree, right? For example, Duchenne's muscular dystrophy, cystic fibrosis, sickle cell anemia. Examples for polygenic include obesity, diabetes, heart disease, right? Such disorders. So, why are none of these statements true? Because they are basically statements which could be true for both a monogenic as well as a polygenic disorder, right? So DNA sequence variation could contribute to disease onset. Yes, it could be either in case of monogenic or polygenic disorder. Then both these disorders can affect either sex, and the affected individuals, they can transmit the disease causing allele to the next generation as well, right? Okay. So moving on to the next question. Okay, so I had actually also included this particular illustration in which basically which says this is a DNA sequence, right? It represents your genome over here. Okay, so if it would be a monogenic disease or monogenic risk factor, then these risk variants would rarely be observed in the normal population, and the binary testing results would be obtained. So what this means, I'm going to explain. So in this case, if suppose you have a genetic risk factor which is a monogenetic disease risk factor, right? So it is the probability of it being a risk factor is very limited, because in case of a risk factor, what happens that it may contribute to a disease. But in case of monogenic disorder, what will happen? There will be one gene that will be responsible for the disease to occur, right? So if this risk factor is present, it will probably cause the disease, right? So there will not really be normal population which will actually have this risk factor. And what does this binary test result mean? That if you have the risk factor, this factor or this risk variant, then there are two options. Either you have the disease or you don't have the disease. That is the binary result, right? So it is either black or white, yes or no, 0 or 1, right? So over here, what does binary testing result means? That in case of monogenic disorders, if you have the risk factor, you will probably get the disease. If you don't have the risk factor, you will not get the disease. It is very highly unlikely, or it is only rarely observed that a risk factor for a monogenic disorder will appear in a normal population, right? On the contrary, polygenic risk is the kind of risk factors that we actually had been discussing about so far. So over here, what it means is that there are these risk genetic risk factors are low risk variants. Over here, it says high risk variants. Means if you have the risk factor, then you will probably have the disease. In case of polygenic, what it means is that if you have the risk factor, you will probably not get the disease, right? There is a good chance that you will never get encounter that disease. So it is a low risk variant. Then numerous common variants are present, unlike this, where in normal population it is rarely observed, right? And there is a continuous testing result. So there will not just be two either yes or no answers, right? It could be in a like in this case, it has a bell-shaped distribution. It has a continuous distribution. For example, somebody who has a risk factor might be 20% at risk of the disease. Somebody might be at 50% risk of the disease. Somebody might be at 100% risk because it has the other factors also, right? So it will be a continuous distribution basically over here, right? Okay. So moving to question number 4. In a typical case control study for gene association, the subject in the case group are expected to differ from the control group in A, age, B, ethnicity, C, genetic makeup, and D, lifestyle. So which one of these are the factors in which the case group and the control group will differ in a case control study? So just think about it. Think about what exactly a case control study is, and these types of studies. I have also made certain slides which we are going to deal with in the discussion presentation, right? So the answer for this particular question is basically genetic makeup. Okay, so what is a case control study? In the case control study, there are basically two groups, as is given in the paper in the question also, which is one group is called case and the other is called control, right? So these two groups, all other criteria are or parameters that are mentioned basically, which is age, ethnicity, lifestyle, they are matched, right? So for example, if there will be a group of people suffering from a particular disease in Africa, so African is basically an ethnicity, right? So the people in the control group will be healthy individuals, which will also be from Africa. They will probably have the same lifestyle. For example, if the affected individuals are farmers, so these control individuals will also be farmers, right? They will be African, they will be farmers, and for example, the age at which these case groups are being studied is about 30. So the control group will also have individuals of about the same age, right? It could be a range of ages, for example, 28 to 32. So these individuals will also lie in the same age range. So why is this done? So that all other parameters are kept constant, so that whatever variation is being basically observed, that we know is because of their genetic makeup, right? There is some difference in their genetic makeup, due to which what one group is basically affected and the other is not. Okay, so moving forward with our fifth question, which says, in the positional cloning approach, the co-segregation of a microsatellite marker with the disease phenotype in an affected family indicates A, that the microsatellite marker is involved in the disease phenotype. B, that the microsatellite marker is located very close to the gene involved in the disease. C, that the microsatellite repeat is expanding in every generation. Or D, that the microsatellite marker is an unreliable marker for positional cloning. So this is a really good question. It tests your concept of whether or not you are aware of what exactly a microsatellite marker is. So just go through the options. I will discuss the solution. Okay. So let us move to the solution part. So it says that the microsatellite marker is located very close to the gene involved in the disease, right? Hold on, guys. Okay. So, what it is located very close to the gene involved in the disease. So, what exactly is a microsatellite? First, let us see that. Okay, microsatellites are also known as simple sequence repeats, and they are typically composed of 1 to 6 nucleotide repeats. So, what does a simple sequence repeat mean? It basically means that the same sequence is repeated again and again, right? For example, if the sequence is A, T, A, T, A, T, then that is how it will be repeated. A, T, A, T, A, T, right? It could be like this two nucleotide repeats. It could again be that uh, ATT, ATT, ATT being repeated, right? So the same sequence is being repeated over and over again for a short stretch of DNA. So that is known as simple sequence repeats, right? And these basically simple sequence repeats or microsatellites are markers. These markers are abundant and they are distributed throughout the genome and are highly polymorphic compared with other genetic markers, as well as being species specific and codominant. Okay. So what does this mean? It gives us some information on what exactly microsatellite is, right? It says, okay, it says that they are distributed throughout the genome. So, for example, if our genome has around 3 billion base pairs, right? So, these microsatellites will be present at some distances throughout, which is distributed throughout the genome. They are highly polymorphic, right? What does being polymorphic means? That it basically varies from individual to individual in a particular population, and polymorphism basically means that it is a genetic variation which basically exists in at least 1% of the population, right? And for example, if there are, what is polymorphism? Let me just clear this out. A lot of people I know have problem in understanding what polymorphism itself is. So if, okay, if these are homologous chromosomes. I'm sorry for their size difference. And this is a particular gene, right? So this is a particular locus at which the gene is present. This is one allele and this is the other allele. In one individual, only two such alleles can be present, right? But what will happen if you consider a population, right? For example, this was one individual. If you see another which has one of the alleles that is represented in orange, and you see a third individual which has one of these repeats, sorry, one of these alleles represented in this purple, right? So what will happen over here is that in this population altogether, there are multiple alleles that are present for this gene. For one individual can have only two alleles, but in the entire population, they can have multiple alleles, right? These can vary in like tens to hundreds, right? The number of alleles, usually in tens. Hundreds is just too much. So such alleles, if they are present in at least 1% of the population, they are known as polymorphic, right? So these alleles will be polymorphic. And being polymorphic basically helps people to differentiate between people from different origins. For example, it is much more likely that the polymorphisms that exist in you and me are comparatively similar with respect to polymorphisms. If I compare me to somebody who was born and brought up in maybe Britain, right? So since the environmental conditions are different, their hierarchy is different, maybe their origin ancestry is different. So polymorphisms will be much more different between me and a person who is born and brought up in Britain than me and maybe one of you, right? But for me, it will be most similar with my family members, right? So, thus, this polymorphism, it really helps uh, in these ancestry studies, and because in polygenic disorders, what happens is that not all genetic variations will be present in all ethnicities, right? There will be some genetic variations that are predominant in maybe Indians, right? So a lot of Indians have heart condition or diabetes, while maybe a lot of French people might not have these conditions, right? I'm just saying hypothetically. I'm not really sure about the rate of diabetes or hypertension in France, right? So these that is why they are saying that these microsatellites are also good markers for disease phenotypes. Apart from these, as where they are also species specific. So I was talking about ethnicity specific, but they are also species specific, right? So microsatellite markers in humans are going to vary from microsatellite markers in mouse, and they are also co-dominant in nature, right? So these microsatellite markers, for example, this was the disease gene that I was talking about, this particular disease gene. So the marker will probably be present over here and over here, right? So these markers will probably, they are located very close to the gene. They will be segregated along with this disease gene. And like what we have to follow is this marker how do we know if this marker is reliable? So we know that by basically seeing that is it almost always, maybe it's at 90% it is present along in that individual which has the disease. So it means that it is almost 90% possible, or 90% of the times it happens that this particular microsatellite is basically segregated along with this particular gene, right? So that is why option B is correct. So along with actually explaining why option B is correct, I got a little uh, in the flow, and I wanted you to understand exactly what microsatellite markers are. I hope that is clear. So we'll move on to question number 6, which basically is again about HapMap project. So they say that in the HapMap project, the term linkage disequilibrium refers to which one of these? Is it the non-random association of alleles at different loci? Is it the random association of alleles at different loci? Is it the linkage of alleles at a given loci? Or is it non-linkage of alleles with a given loci? So what does linkage disequilibrium mean? So it has 2 or 3 confusing options, but we'll see the solution. We'll try to clear it up, right? Okay, so the solution for this particular question is the first one, right? So linkage disequilibrium, what does it mean? That there is a non-random association of alleles at different loci, right? So, do you remember the Mendelian genetics? In the Mendelian genetics, it says that the association between alleles at different loci is basically random, right? Or a lot of the primitive genetic concepts that we have studied say this, right? But it is not always true. And the HapMap project, when it gave the data, this particular term was basically coined, which means that there is non-random association of alleles at different loci. What does it refer to? So LD refers to non-random association of alleles at a pair of genetic loci. It manifests as a deviation of observed haplotype frequencies from the frequencies expected under assumption that alleles at two loci associate independently, right? It also says that it is a population-based parameter that describes the degree to which an allele of one genetic variant is inherited or correlated with an allele of a nearby genetic variant within a population, right? So what happens is that why do we need to study this particular term linkage disequilibrium? Because when the Human Genome Project data came along, we also got to know about one term which was linked genes. So what were these linked genes? The genes that tend to be basically carried onto the next generation together, right? So there was no crossing over between these genes. But it was along with this linked gene concept, when the HapMap project was basically studied, there was another concept that came along, which was linkage disequilibrium, which said what? That earlier it was expected that alleles at two different loci associate independently. There is no correlation between the alleles at two different loci. But when we actually studied the HapMap project, sometimes what happens was that there were interactions between such different alleles which were present at different loci, which basically gave this new concept, which was known as linkage disequilibrium. So I did not really find a very good illustration. I wanted to show exactly how this pictorially represent this concept, but that I could not find. If I find, I'll include it in my presentation before uploading it, right? So, okay, question number 7. The term haplotype refers to. So, this is a concept that we dealt with in our discussion our last week, right? I hope people who were present, at least the people who were present would know it, and the others too, right? So it says a group of individuals testing positive for a specific antibody or a molecular biology method used to track the inheritance of genes in families, or a set of markers that are physically linked and segregated together, or markers representing haplotype genome. So which one of these uh, is basically kind of a definition of the term haplotype? Okay, so let us see what the answer is. Okay, so it is basically a set of markers that are physically linked and segregated together. So this was the same illustration that I used last week in the discussion hour, right? So what happens is that in a particular individual, let us first see only individual one. In this one individual, what happens is it has different single nucleotide polymorphisms. What does SNP stand for? Single nucleotide polymorphisms. Again, the same polymorphic phenomena that we just discussed. So what happens is that at different base pairs, it has these SNPs, right? And the SNPs that are present in this one individual will differ to some extent from the SNPs that are present in this particular individual, or SNPs present in this particular individual, right? So when you actually link these particular physically link these particular haplotypes or this particular SNPs, right? Like what is done over here, you basically obtain something which is known as a haplotype. And how, what is a characteristic of this haplotype? Only the SNPs that will physically, that will basically be segregated together will be linked over here, right? So what does that mean? It basically means that this TGTCT will probably, when mitosis and meiosis, when meiosis occurs, there will be no a break or a recombination between this particular region from here to here, and these particular SNPs will be segregated as one particular unit, right? That is why we are listing it as a haplotype. Similarly for individual 2 and individual 3. Okay, guys, just give me a moment. Okay, I hope I'm still visible. And my screen, I don't think my screen is visible, right? I'll just share again. Okay. Okay. Cool. So I hope you got a little bit more time to actually go through what this particular illustration, because I find it to be a very relevant to actually very relevant to actually understanding what a haplotype is. This is what actually cleared my concept in the first place, and I was struggling to understand what a haplotype is. So, okay, moving on to the next question. It says, just a moment. It says, while advances in genomic methods have helped us identify the genes mutated in the genetic disorders, the challenge has been to dissect the mechanism by which the affected gene causes the disease. Right? Which one of the following options offers an explanation to this challenge? Okay, so what it asks is, what is the challenge which is still present between us and identifying the mechanism by which a particular uh, disease is caused by a gene, right? So the first option is the physiological function of most of the genes are unknown. B, most of the human genes remain uncharacterized. C, there are not enough biochemical assays to evaluate the function of all genes, or D, all of these. Hold a moment. Hold on. Okay. Yeah. So, yeah, we've gone through the options for this particular question. And I think this is a relatively simple question for anybody who has attended Sir's lecture because it is a very direct question from one of his slides. So we'll not waste much time. We'll just come on to the solution. And the answer for this is basically all of these. And I think it is pretty self-explanatory. Again, as I said, anybody who has actually gone through all of Sir's video lectures, this has been picked from one of his slides, right? As it is, it has been picked from there. So these are the challenges that are ahead of us, because of which basically this we have not been able to dissect the mechanism by which affected genes cause the disease, right? So this is still a very difficult question to answer just because of these challenges, which are pretty huge challenges. So we as basic biologists are researching just to answer such questions. So moving forward to question number 9, which says two distinct genes that are likely to be inherited together are called which one of these? Alleles, polygenes, linked genes, or none of these? So which one of these do you think is the correct option? While discussing one of the questions, I think I've already given away the answer. So we'll just see what it is. So the answer for this particular question is linked genes, right? So what are linked genes? If two genes are located on the same chromosome and in close proximity, right? So one, they should be on the same chromosome. Second, they should be at close proximity, right? So if they are on different chromosomes, they will not be linked, or if they lie very far apart, they will not be linked, right? Why? Because then if they are present in close proximity, they tend to segregate together and do not obey the law of segregation. So this law of segregation, which was one of Mendel's three laws, it is not obeyed by these particular set of genes which lie on the same chromosome and lie in very close proximity. Hence, they are basically known as linked genes, right? So law of segregation basically used to say that any two different genes will basically segregate independent of one another, right? Why? Because if suppose this in this particular example gene 1 and gene 2 were present, what would happen? There could be a recombination at this loci, at this loci, at this loci, at so many places between these genes, right? And if that say a recombination or that break would occur over here, then this gene will probably go in another chromosome and this will go in another chromosome, right? So they will still not remain together. Or if they are in different genes, obviously this has bound to happen. This phenomena will obviously occur. But if they are at they lie very close to each other, then the probability of them having this recombination at this particular loci is very less, right? It is almost next to negligible, right? So that is why these two genes, which lie very close by, will always move on to, after recombination, will always move on to the next chromosome together. So they will be linked. Okay. So the 10th and the final question for this particular assignment was, which one of the following statements is true? First is all DNA variations are mutations. Second is all mutations affect survival. Third is all variations are in intergenic regions. And D is some variation may increase fitness. So which one of these statements is actually true? So people who have been attending my sessions also and who have been actually watching sir's lectures also, this was the concept that was with in the first week, right? And I also basically dealt with the differences between mutations and variations. So that gives away that obviously it is not the first one. So what is it? That the true statement among all of these is that some variation may increase fitness, right? So that is also one of the differences between mutation and variation, right? So just to illustrate this, I have put in this particular representation. First, let us read what variations are. So variations are basically provide raw material for evolution, which may turn out to be useful for the organism. That is why other options are incorrect over here, right? So in this particular illustration, what happens is that, for example, this particular orange color, I'll just call this color orange, this color red, and this color yellow, right? So that nobody gets confused. I've just pointed it out, right? So this orange color was the normal that was existing in the ancestors. When this ancestor basically they basically procreated, their progeny had either this orange color or this yellow color or this red color. So among this yellow color one would be referred to as a mutation because it was later unfavorable, because of which this particular individual or its progeny died, right? This orange one was still getting sustained, and this red one was a new variation which was also getting sustained in the later generations, right? So after mutations created a variation, the unfavorable mutations were selected again. So that is why this yellow one, the individual is now dead, right? The person who contained this particular mutation or variation has died. And the other two, they basically reproduce. When they reproduce, and new mutations might occur, right? For example, there are no mutations shown in this particular orange one, but in this red one, there is one other mutation giving this maroon or brown phenotype also, right? So after that, maybe the environment in which these four individuals were staying at changes, right? For example, first they used to stay at some place which was a plain, and all of a sudden maybe this global warming hits, or this ice age global warming phase, it has been going on for like millions of years now, right? So maybe one of these global warming curves hit, and it is very severe. There is probably a drought everywhere. So what happens, the variations that were carried in these particular individuals which were orange, they were not good enough for them to survive, so they also perished. And these new variations that were created over here and over here, they were more favorable mutations and they were more likely to survive, right? So they survived and they reproduced further. So this particular variation, it perished altogether as this variation. But this new variation, it actually survived, right? So that is how variations provide raw materials for evolution. This might have been a very primitive kind of human, maybe um, Homo habilis, right? And this might be this could be a maybe Neanderthal, and this could be a Homo habilis, right? So continuously there is variation that is going to be taking place, uh, which and some variation will always increase fitness, right? So as to lead to evolution. So that brings us to the end of our first presentation, which was the previous year assignment for week 4. Um, okay. So that is it. Let me just stop sharing my screen. Okay, I've already stopped. Okay, hold on, guys. I'm not sure if there is a problem with my network. There is somebody I'm trying to admit, and that I'm unable to. Okay. Okay, there's this one individual I'm unable to admit him to the meeting. Anyway, we'll just move on to our next presentation, right? So we have ample of time. I think I'll be able to finish that. Okay. All right. So is my screen visible to everybody? Okay. I will just share my screen again because um I have actually just for this purpose I have also joined the meeting from an external link and I can't see my screen over there. Right. So I'll share again. Okay. I'm so sorry, guys. I'm having some trouble with the internet and I'm not sure whether or not my screen is visible. Maybe if one of you could tell me. Okay, I got a thumbs up. That means it's visible. Okay. So I'll continue with the presentation then. In case somebody is unable to view it, please let me know. Okay. So for week 4, as I already told the people who had joined at the beginning, but I also received a lot of requests during my session. So I'm not sure if everybody was there when I actually discussed about, just a moment, when I actually discussed about what I'm going to talk about in this particular presentation. So, since the concepts were relatively less in the final week, and most of them had been either taken up by me already, and not many new queries were there in the discussion forum also. So, the concept part is very limited in this particular week's slides. But instead, what I've done to cover up for the time that we have is that I've added restriction mapping questions right at the end. So that is something we are going to do after we discuss the few concepts that were dealt with in this week that people also had problem with, right? So, okay, we'll just be moving forward. So our first Okay, so this was actually a query on the discussion forum about what exactly is a genetic marker, right? And how can you only amplify a genetic marker? And I do not really remember about who particularly the person was who had this query. But over here, I've included this definition of what a genetic marker is and also along with it, a very basic illustration, right? So just let me get the pointer right.
Hello.
So what is a genetic marker? It is a DNA sequence with a known physical location on a chromosome. ठीक है? So this is the first characteristic of a genetic marker that the physical location of this particular marker is on a chromosome is already known, right? That is why it will act as a marker. What is a marker? It is basically going to be kind of a landmark on that particular genome chromosome, right? For example, if someone was visiting my house, right, and it is difficult for me to explain exactly where my house is if I don't use landmarks, right? For example, if this particular look, sorry,
You're not.
if my house was at this particular location on this particular road, and around it all the other buildings were also houses, right? So
Okay.
these lines, they represent other houses. So what will I say? I might say that okay, my house is green in color, or the roof of my house is maybe conical, but the others around me have a flat roof, right? So these might be certain characteristic marks of that person, uh, uh, for that person to actually locate my house. But what if this person was not from this particular city? Okay, for example, okay, Ricky, do you have a problem?
Yeah, you are not have
Do you
like
have a question?
you're not visible on the
You
screen.
can speak up.
Yeah, you're not visible on the screen. Hello.
Speak up. Okay, just a moment. Now you can speak up.
Yeah, you're not visible on the screen.
Oh, I'm not visible. That was actually one of my issues. Okay, just a moment. Okay, now am I visible?
Yeah.
Okay. All right. I'm so sorry to waste maybe 5 minutes. I had just read the first statement over here about what a genetic marker is. So the first statement said that it is a DNA sequence with a known physical location on a chromosome, right? Okay, so let me read the entire thing, then maybe I'll explain what it is. Apart from this, genetic markers can help link an inherited disease with the responsible gene, right? DNA segments close to each other on a chromosome tend to be inherited together, right? So this is one of the phenomena we have been talking about. Genetic markers are used to track the inheritance of a nearby gene that has not yet been identified, but whose approximate location is known. The genetic marker itself may be a part of a gene or may have no known function, right? Okay. So what we can draw uh, an analogy, right? In this analogy, what we say is that this particular genetic marker is a landmark, right? So for example, a person who is maybe I presently am in Kanpur, right? So I want to tell another person who is also in Kanpur about where exactly my house is, right? So in order to tell this person, I need to tell him or her certain landmarks, right? Or for example, if I was at the institute, okay, at Indian Institute of Technology, Kanpur, I want to tell him where my lab is located at, right? So what I will tell this person, I will tell this person to first locate, maybe just a moment, to first locate Indian Institute of Technology, right? It is a relatively larger place. It is easier to locate, right? So this person who does not know me, who does not know where my lab is, I will ask this person to locate IIT, right? IIT, so that this person can basically ask somebody, okay, where this particular institute is, and this person might be able to reach to this larger destination, right? Now, among this 1,000 acres, I want this person to find out where I am located right now. Right. So, for that, what will I do? I will basically use certain landmarks that are very famous in this particular IIT. So, why will it be a landmark? Because these are places that are relatively famous. People usually in IIT or people who visit IIT often usually know these destinations. Okay, for example, I can tell this person to locate the famous library at our institute, which is PK Kelkar Library, right? So I can ask this person either to locate PK Kelkar Library, and from there I can tell this person, okay, you have to move north from. Once you find this library, you have to move north and then you will find a building that is maybe round in shape. You have to locate that building. That will be the tutorial block. Ahead of that, you will find the Department of Biosciences and Bioengineering. That is where I am going to be, right? So this could be one genetic marker. Or this could be one landmark, right? It could act as a genetic marker for that person to locate a particular gene, which is what? Which is me or where my lab is, right? Another one could be, so why am I taking another example? Because these genetic markers could be more than one, right? So what could be another genetic marker? Maybe, okay, so there is this very famous crossroad at IIT Kanpur, which has a statue, and this particular crossing is known as Kargil, right? The statue is known as Kargil. So I can ask this person to locate that Kargil crossroad and to walk straight from there and take the second right, right? So if that person takes the second right, after walking straight from Kargil, again, that person will arrive at my department and that is where I'm going to be. Right. So it is another genetic, it could act as another landmark to basically locate my particular position. So, what are the characteristics of this landmark? That these landmarks should be very well studied or understood, right? In order for them to act as a landmark, they should be very well understood. For example, if you are located in a residential colony, it is difficult to navigate, right? For example, if you ask a Swiggy or a Zomato delivery person, or if you are making them understand where they actually need to reach, if the GPS network is not so good, then it will be easier for you to say, okay, I am located next to Indian Overseas Bank building, than you saying that there is this white color house next to it, there's a green color house next to it, there's a house where I am sitting, right? So that is more difficult than if you actually have a landmark that is very well known. For example, an Indian Overseas Bank in your locality. So that is how this genetic marker works, right? So in this illustration, we say that, for example, there is this BT gene. For example, the gene that was one of the first recombinant crops used in one of the first recombinant crops in cotton, right? The BT cotton gene. So if this BT gene is present over here, how do we locate this gene? We know that it is somewhere on this particular chromosome, but how do we know where it is? So, for example, whenever we have sequenced this gene, we have always found this particular marker along with it, right? So now we know that if we basically tag this marker, this gene will be maybe in its proximity only, right? So we need to find one marker maybe over here and one marker that is maybe upstream of this gene, so that if we tag these two markers, we will definitely get this gene also, right? So that is how these studies are performed, and that is why this genetic marker is so relevant. So moving on to the next slide. Okay, somebody also, we have discussed this multiple times about what Angelman and Prader-Willi syndrome are, but usually we have discussed it with respect to these two disorders, that is uniparental disomy and imprinting disorders, right? So we have never really gone through what exactly these disorders do, right? So in Prader-Willi syndrome, we are actually, I have a slide in which we will discuss what exactly these disorders do, what are their end effects or end results. But first of all, let us revise our concept. Prader-Willi syndrome, we had studied it in case of maternal uniparental disomy, right? If there is maternal uniparental disomy, it is likely that the a child that actually exists with this maternal uniparental disomy is going to get this Prader-Willi syndrome, right? Or if there's paternal uniparental disomy, again, it is likely that the child suffering from it is going to get this Angelman syndrome, right? But the chances are also listed over here. So if there's paternal deletion, then there's of this particular chromosome, which is chromosome 15, right? Both these disorders lie on chromosome 15, Q arm. Hold on. Okay, so both these disorders, they lie because of defects on chromosome 15's Q arm, right? So if there's a paternal deletion in this particular Q arm of chromosome 15, then there's 70% chance that Prader-Willi syndrome will occur. If there's maternal uniparental disomy, 28% chances that this defect will occur. And if there's an imprinting defect in this, that is, if there's abnormal silencing of the chromosome that should remain active, then again this Prader-Willi syndrome will occur. Similarly, for Angelman syndrome, 70% chances are there if there's maternal chromosome deletion. 2% chances of paternal uniparental disomy is present. Imprinting defect, if it is present, 2 to 5% chance for Angelman syndrome to occur. Then this particular gene mutation, 5 to 8% chance is there for, yeah, Ricky.
Yeah, again, the visibility went off.
Okay. The screen is also not showing.
Yeah, everything is blackout.
Is it better now?
Yeah, now it is visible.
Okay. All right.
Again, it went off.
I'll just present again. Okay. I hope now it is visible.
Yes.
My screen is also visible now.
Yes.
Okay. All right. So these are certain causes of Angelman syndrome. It could be either due to maternal deletion, paternal uniparental disomy, or imprinting defect, or due to mutation in this particular gene, which is UBE3A. Or there was also a 12 to 15% chance of Angelman syndrome occurring, the cause of which was unknown, right? So it is still unknown. So that has been marked as question mark over here. So these are basically the causes of the Prader-Willi syndrome or Angelman syndrome to occur, right? So now somebody wanted to know what exactly the syndrome is. So for that reference, what happens in this Prader-Willi syndrome and this Angelman syndrome is that, okay, first thing you can remember is that Angelman comes from the name, right? You can deduce something from its name, that is angel-like, right? So, the children who actually have the syndrome, they basically are they are have severe mental retardation with respect to children who have Prader-Willi syndrome, who have mild or moderate mental retardation, right? But these children who have severe mental retardation and Angelman syndrome, they often have this one characteristic that they will usually it will either appear that they are always smiling, or they will actually always be smiling, and there will be these paroxysms of laughter, right? Which means that there will be these sudden bouts of laughter that will occur, right? So they will appear very um, angel like. That is why the name Angelman syndrome. On the contrary, you can remember the children who are a little grumpy and who do not really have this paroxysms of laughter, they will be the Prader-Willi syndrome children. So, the certain other characteristics for Prader-Willi syndrome are that these children will have feeding problems, they will have neonatal and infantile hypotonia, right? For example, what is hypotonia? That their muscle tone will be reduced, right? Then hyperphagia, that they will always feel very hungry. Then they'll be obese. They will have genital hypoplasia. Their genitals will basically not be very well developed, right? Then they will have certain facial characteristics which are dolichocephaly. Um, Okay, so what happens in dolichocephaly? It is basically, I think I'll have to draw, and I'm still not sure if I'll be able to explain. Okay, what happens is that, for example, if we see a human being or a person from the side, they are probably going to have this shape of the skull, right? Sorry, this shape of the skull. Okay, you will not be able to. Okay, so I don't have a very good writing surface on. So if this was the nose, this was the mouth, right? So this will be the shape of the skull normally. But in people who have this dolichocephaly, what will happen? That if this was again the nose and the mouth, then their skull will be like this, right? So it will be expanded in this way, and it will look very weird because it will be really expanded uh, anterior to dorsal end, right? It will be really elongated. So that is what dolichocephaly means. Then they will have a narrow bifrontal diameter. What does that mean? That this particular this particular diameter will be very reduced in these individuals. So the face like this will be very compact, but like this it will be very elongated, right? Uh, okay. Then they will have almond-shaped eyes, small appearing mouth. Their mouth will be very small and downturned corners of the mouth. They will usually have this corner downturned, right? Then they will have certain behavioral problems, short stature, small hands and feet, and hypopigmentation. Right. So these are all self-explanatory points. What will happen in Angelman syndrome? These children will have severe mental retardation. They will have ataxia, that is basically difficulty in movement. I I actually just have to verify what ataxia is. I should not be telling something incorrect. So impaired coordination of your like your movement will be impaired because there is impairment of the coordination, neural control of coordination, right? Then these children will be very hyperactive. They might have convulsions, which will basically look like seizures, right? Because their muscles they will tense and relax very rapidly. So they will kind of have these convulsions or seizures. Then they will have the paroxysms or bouts of laughter. Then speech will almost be absent. Their movement will be clumsy and jerky. Then their facial characteristics include microcephaly, that is, their skull will be very small, right? For example, if this is the normal skull, then their skull would just be this much, right? So it will be a very small skull. Then prognathism, which is basically that the lower jaw, it will basically be protruding outward. Right, then macrostomia or a large mouth. Sometimes what happens in these children is that their maxillary and mandible um, muscles they are not fused together, so their mouth is actually enlarged, right? Because that fusion has not occurred. So macrostomia will occur. Their tongue will be protruding and their teeth will be widely spaced. Then there will be ocular hypopigmentation, that is less pigmentation in the eyes, and abnormal EEG. right? So these are certain characteristics for Prader-Willi syndrome patients and Angelman syndrome patients, both of which are defects in the long arm of chromosome 15, right? As has been discussed in the previous lectures. Okay, so another thing that I wanted to discuss was study designs, because I feel that um, now if you are actually coming across these case control study or maybe retrospective cohort study, then maybe you should also get to know what kinds of study designs are actually there, right? So I will start from the broader kinds and then I will narrow it down. So the broader kind being either cross-sectional study or a longitudinal study. So these are certain study designs. Okay, first let me explain this particular illustration. So over here, past, present, and future represents the time, right? The timeline. And these are several study designs, for example, cross-sectional study, prospective study, retrospective cohort study, case control study, or randomized controls trial, right? So these are particular kinds of study designs. And the arrow over here and where it is actually lying basically represents that what aspect is being studied. For example, in retrospective cohort study, only present and past is being studied. In case control study, only present and past is being studied. Why is this so? We will actually understand when we are discussing individually about these particular study designs, right? So the first design being cross-sectional type of study, right? So what is a cross-sectional study? Just let me have water for a moment. Okay, so cross-sectional study is a type of research design in which you collect data from many individuals at a single point in time, right? In cross-sectional research, you observe variables without influencing them. So there are two takeaways from cross-sectional study. One, it is basically the data is a snapshot, right? Because at a single point of time, you have collected data from different individuals. So it is a snapshot. Also, it is just an observational study. You are not going to basically influence the individuals in any way or change their environment and study them. You're not going to do that. It is just an observational study of the data that is collected at a single point in time, right? So, okay. Okay. From this illustration, if this is the time axis, at a single point, there was data collected from different kinds of individuals, right? So, and then this data was basically observed for differences, variations, etc. So, such a study is known as a cross-sectional study. On the contrary, there is another kind of study which is basically known as longitudinal study. So, what are these longitudinal studies? These basically employ continuous or repeated measures to follow particular individuals over prolonged periods of time, often years or decades, right? So let me just put its illustration also, right? So over here, if this is the time axis, then at various time points, these particular individuals will be studied, data will be collected from them over maybe years or decades or maybe just weeks or months, right? But over different time points. Right. In this case, such a study is known as a longitudinal study, and it is clearly different from a cross-sectional study, which is just a snapshot of a particular time point. Right. Thereafter, let us understand the types of now. Okay. Okay, going back. I'm unable to go back actually. Huh. Going back, uh, cross-sectional study was just this particular type of study, right? And longitudinal study was a depiction of all the kinds of studies that we are going to deal with ahead of this, right? So, just a moment. Okay, so ahead of this, the cohort studies that we are going to deal with, the case control studies, or even the uh, What was the last one? The trial study, right? All these studies are going to be a part of the longitudinal studies because it is done over a period of time, right? So, the cohort studies, it can basically be divided into two types. One is a prospective cohort study, and the other is a retrospective cohort study, right? So, if you just go by the terms, prospective is something that could be, retrospective is to basically reflect on what has already occurred, right? So, what is a prospective cohort study? It is a longitudinal cohort study that follows over time a group of individuals who differ with respect to certain factors under study to determine how these factors affect rates of certain outcomes, right? So, in this, we are going to take a group of individuals. We are going to study them over. Okay, we are going to take a group of individuals who are very similar, right? The things that we discussed in our question answers, that they will probably have the same lifestyle, they'll probably lie in the same age group, and from the same ethnicity, right? So all these things will be similar among these individuals, and then they will basically be kept or given certain similar factors, and then they will be studied on how these particular factors are basically affecting the rate of outcome in these group of individuals. Now, what is a retrospective cohort study? It is another longitudinal cohort study in which the medical records of groups of individuals who are alike in many ways but differ by certain characteristics are compared for a particular outcome. This particular type of cohort study is also known as historic cohort study, right? So in this, what happens is that up till now, we will basically get a similar group of individuals, and we will study their historic record, right? Or medical records, that whether these individuals who are very similar otherwise, how did some of them have cancer, some of them do not get cancer, or something like that, right? So this is an illustration that I took out from Wikipedia, basically, which basically says the first one is a prospective cohort study, which says that the there was already exposure, right? These are there are these similar group of individuals who were under similar exposure or similar conditions, and then we basically chose these particular individuals, we started studying them. So this is basically where the research started, right? This particular man depicts where the researcher came in and where the research started. And now this researcher is going to study these groups of individuals for some time, maybe weeks, months, years, decades, to see how maybe disease is being caused in them, or what is the kind of disease if suppose both of these individuals have cystic fibrosis and both are of a similar genetic background or similar background, maybe not genetic background, both are from a similar lifestyle, age, background. So this doctor intervened and started basically studying these individuals, and now this doctor wants to see that maybe after 1 year, how does disease progress in this particular group of individuals and this particular group of individuals, right? So what is the differential progression of disease? Maybe that will give them some answers of how their genetic background might have been a little different in order to lead to such differing or differential disease conditions, right? Similarly, what is a retrospective cohort? That maybe there were these two individuals who had cystic fibrosis and they developed the disease. Now, these diseases were manifesting a little bit differently in both these particular individuals. And what happened was that after this manifestation was already seen and both individuals were being treated, this scientist or this researcher came in. And what this researcher said was that, okay, let me study their medical history in order to see that how these two very similar individuals have different disease manifestations, right? So if the intervention is before the disease properly manifests, right? That will be a prospective cohort because they have to look into future, like they'll have to keep addressing these particular patients in order to find how their disease is going to manifest. And if the disease has already occurred, right, the manifestation has already occurred, and later the research angle comes in, then it is going to be a retrospective cohort, right? So I hope this is clear. Moving on to our next kind of study, which is basically a case control study, right? So in a case control study, we will compare two groups of people, those who have the disease or the condition under the study, which will be the cases, and very similar group of people who do not have the disease or condition, which is controls. So this I have also explained while we were going through the PYQs, right? So what will happen in these particular individuals is that histories of these individuals will be taken. For example, the disease condition that we are studying is cancer, right? And the control individuals are the individuals that do not have cancer. So the patients with cancer, the histories will be taken. Similarly, the histories of the patients without cancer, which is the control group, will also be taken, and then their histories will be compared, right? And then conclusions will be drawn based on that. So such type of a study design is known as a case control study. Now, the next kind of study is basically known as a randomized controlled trials, right? In this, uh, RCTs are prospective studies that measure effectiveness of a new intervention or a treatment. So sometimes what will happen is that, for example, a drug has been introduced, right? So it is still at that clinical trial phase, right? You have heard of drugs or injections or therapies being at clinical trial phases, right? So in these phases, what will happen? These kind of studies, which are known as RCTs, in which basically there will be a set of patients that will be taken, right? So in this, there are no normal individuals, there are just patients, right? So maybe all these patients are suffering from cancer again, right? So all these are cancer patients. Now, randomly, these patients will be divided into two groups. One group will be the treatment group and one will be the control group. So now how this random assignment is done is basically through computer algorithms, right? So if the computer is basically separating these individuals, and if it is a blind research, so the computer will separate the individuals, and even the researchers will not know which one is the treatment group and which one is the control group, right? So if they do not know which one is which group, then there will be lesser bias in their observations, right? So now these patients have been divided into treatment and control group, and then one will treatment group will basically be given the therapy, control group will be given the placebo, and or just the vehicle, and then follow up will be done with these individuals, their other environmental conditions, the kind of people that they are meeting in the hospital, all these are going to remain constant, and for both the groups, right? Then both the groups will be followed up and their results will be compared, and at last it will be revealed which individuals were in the treatment group and which ones were in the control group, right? So that is how basically these randomized control trials are done. Why will it help? Because sometimes what might happen is that the treatment might be very good for the patients, or on the contrary, what might happen is that the treatment might even be toxic for the patients, right? So in order to understand how it is basically what is the effectiveness of the new intervention or treatment, types of study designs are basically put through, right? So moving forward, okay. Now, next I wanted to actually discuss omics. Just let me see what the time is. Okay, it's almost 4:30. I will just, anyway, I had not prepared very concrete slides for omics, right? I just wanted to discuss the concept with you guys. So I'll maybe rush through these slides so that we can solve those numericals that I have, right? So, what are omics? So, this slide I have just put in to tell you that there is just not genomics and transcriptomics and proteomics, right? There are many more kinds of omics that have emerged in recent years. So, this is just a representation of all those, right? These are certain major ones. These other ones are still under emerging fields, right? So, but there are different kinds of omics, right? Let us move on to the next slide to see what are some major types of omics and what they will basically be doing, right? So the most primitive one was genomics, right? Now what it does is structural functional analysis of genes and identification of genetic variants. Then comes transcriptomics. Transcript basically means mRNA, right? So over here, gene expression profiling is done, functional regulations are profiled, and differential expression of these genes are checked through transcriptomics. Then comes proteomics, which is basically study of proteins. So protein identification, quantification, and post-translational modifications are studied over here. Then this is a very emerging field right now, which is metabolomics. Metabolomics along with lipidomics, in which basically the lipid profile is studied. But in metabolomics, there is profiling of metabolites, hormones, and signaling molecules. Nowadays, it is actually being used by a lot of labs to actually um, reduce the research and like the basic biology research and the clinical intervention distance, because what happens is that like the gene is on how do I explain? Let me see if I have not put in that illustration. Okay. So what happens over here is that if you see a gene, an mRNA, a protein, and metabolites, right? So metabolites are byproducts of proteins also, right? So they come even further after proteins. So genes are not directly related to the phenotype of an individual, but proteins can be directly related to the phenotype of an individual. So what happens sometimes is that even if there is some variation that we find in genomics, it might not be translatable as a variation in the proteomics data, right? So it might not be carried forward to the protein level. Similarly, if the concept nowadays being followed is that if there is a variation that could be found in at the metabolomics level, then it will be much more helpful for the patients or the population in general, because such variations, they are directly correlating to the phenotype of the person, right? So if a particular metabolite is very significantly upregulated in one particular person as compared to the normal population, then this particular upregulation of metabolite might be the contributing agent of the particular condition that this patient is suffering from, right? So it is very close to the phenotype. So that is why proteomics, metabolomics, these are even more up and coming fields as of now. So phenomics is basically morphological, molecular, physiological, and chemical biochemical characterization, and ionomics is identification, characterization, and distribution of elements, right? So okay, this is just a slide that I have mentioned so that you guys have a reference of what strategies are used for different types of omics. For example, maybe whole genome sequencing and whole exome sequencing for genomics. Then for epigenomics, maybe this ATAC-seq, which is one of the latest ones. Then for transcriptomics, RNA sequencing, maybe, right? For metabolomics and lipidomics, mass spectrometry, NMR spectroscopy. So these are the kinds of approaches that are used to study these particular omics. So just for your reference, I have basically put in this slide. You don't really need to mark up all this. You will eventually learn when you start reading research papers, right? So just for your reference. And what are the broad applications of omics? So it could either lead to the discovery of certain different like discovery of certain new genes or mRNAs or maybe metabolites that are involved in a particular disease, right? It could lead to better diagnosis. How? Because maybe if one protein level is more differentially, if there is much differential uh, upregulation or differential down regulation in any of the genes or maybe at mRNA level or protein level in one particular gene, then it might lead to act as diagnosis for a particular condition. Then it also helps maybe the doctors or the clinicians to decide what kinds of therapies should be given to the patients. For example, if you say that a particular individual has like a particular type of metabolite due to which a particular drug will be toxic when it interacts with that metabolite. So since this metabolite is very highly like it is present in a very high quantity in a particular individual, maybe this kind of drug or this class of drug should not be given to this individual because it does more bad than good, right? So such kind of decisions could be made by the clinicians based on such data. Okay, I have basically rushed through those slides so that I could reach over here. So the first thing that I wanted to do, the first question was basically this. So if somebody says that ECO R1 and RSA1, these are basically two restriction endonucleases. So if ECO R1 and RSA1 restriction endonucleases require 6 base pair and four base pair sequences respectively for cleavage, right? In a 10 kilobase pair DNA fragment, how many probable cleavage sites are present for these enzymes? Okay, so this is something that we are going to solve. I will just have a water break, the last one probably. Okay, so just a moment. Okay, I had a bout of cough. I did not want you to bother you. Huh. So let us solve this numerical. I don't expect you people to know the all the answers, but what I'm here for is for you to basically address different kinds of questions, so that if in future you basically come across such questions, you will be able to answer them, right? So let us move towards the solution. I don't know if I've put it or should. I've put a little bit of it. Okay. So that actually is for your reference. When I when I upload this presentation, this solution is for your reference. What I can do is I can basically explain you right over here. So what happens is that we say, or maybe that is a better illustration. So eco R1 requires a 6 base pair sequence and okay, I'm not visible again, Ricky.
Yes.
Okay, just a moment. Am I visible now?
No.
Let me just escape from. Hmm. Visible? Okay. All right.
It went off.
So it went off. I have no clue why this is happening. Okay, I don't think it's visible still. Hold on. Just a moment. Is it visible now?
Yeah, as of now it's visible.
Now it's visible. You can see my screen moving also. Okay. I will try not to disturb it now. Okay. So over here, I hope the question everybody has read that EcoR1 and RSA restriction endonucleases we are talking about, and the question says that EcoR1 restriction endonuclease requires 6 base pair sequences and RSA1 requires 4 base pair sequences for cleavage, right? So if we draw it, like it is just a normal question. We do not need to remember the restriction sequence for EcoR1 or RSA1 in this particular example, right? So we can just say that in EcoR1, 1, 2, 3, 4, 5, 6, 6 base pair sequences present basically, right? It recognizes a 6 base pair sequence where it will cleave, and RSA1 will recognize a 4 base pair sequence where it will cleave, right? Now, what else does the question says? It says that there is a 10 kb DNA fragment. How many probable cleavage sites are present for these enzymes? So these are base pairs. This is base pair and this is kilo bases. Right. So 10 kb would basically mean 10 into 1000 base pairs. Right. So that if those many base pairs are present, which will basically mean in 10,000 base pairs, how many probable EcoR1 and RSA1 sites will be present? Right. For this, first let us see that what is the probability for each base pair in EcoR1. Right. So for this particular base pair, what wait, this particular base pair could either be A or it could be T or it could be G or it could be C, right? So for it to be any one of these, the probability is 1 by 4, right? Similarly, for this base pair to be any one of these, the probability is again 1 by 4. So similar is true for all 6 of them, right? So the probability for 6 base pairs, which is basically the recognition site for EcoR1, which is a proper one particular sequence of bases, right? For that particular site to be in this particular 6 base pairs, what is going to be the probability? It is going to be 1 by 4 into 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 1 by 4. So 1 by 4 raised to power 6, right, is going to be the probability. So I calculated it to be 1 by 4096, right? Similarly, for RSA1, if the same concept is applied, what is going to be the probability for each of the base pairs? It is basically going to be 1 by 4 into 1 by 4, that is 1 by 16, into 1 by 4, 1 by 64, into 1 by 4, 1 by 256. So 1 by 256 is going to be the probability for RSA1, right? Now it says that in 10 kb fragment, what is going to be the probability for this to occur and what is going to be the probability for this to occur? So in 10 kb fragment, how many base pairs are there? 10,000 base pairs. Okay, 10 into 1000 because it is a kilo base pair, right? So what will be the probability of eco R1 to occur? It will be 10,000 into 1 by 4096, right? And probability for RSA1 will be 10,000 into 1 by 256. So, okay, I don't think I've actually finally calculated this. Uh, Hopefully you can see my calculator too on the screen, right? So let us do this. 10,000 divided by 4096 is about 2.5, right? This is around 2.5. We will see which answer is the closest to this. Okay. And what is the second one? The second one says basically 10,000 divided by 256, right? It is going to be 39, around 39. So let us see which one of the, let us see which one of the examples is correct. So, okay, 2.5, the closest one over here is 2 only, right? And 39, the closest one is 39 only, right? So this is the correct option for this particular question. I hope it was understandable for all of you, right? If at all there were any problems, you might tell. Otherwise, the solution that I have mentioned, the slide will be available, right? I have just not calculated this final thing because it is anyway marked over here, right? Moving on to the next question, I actually have two more. This is the second last question. This question says that draw a restriction map site for a linear vector when the restriction digestion yields the following results, right? So it says that if there is a restriction digestion by eco R1, then a 3 kb fragment and a 3.5 kb fragment will be received. And if there is a restriction digestion by hind 2, then a 2 kb fragment and 4.5 kb fragment will be obtained, right? If there is a double digestion, what does this mean? It means a double digestion by both eco R1 and hind 2 is occurring, then there will basically be three fragments that are obtained. One is 2 kb, one is 1 kb, and one is 3.5 kb, right? So using this particular information, what we have to do is that we have to draw a restriction site map for a linear vector when the restriction digestion yields the following results. Oh, I just read the entire question, not necessary. Okay, so how will we do this? First, we have to keep two things in mind. First, we have to find the additive regions between single and double digestions. Secondly, we have to find the overlapping regions. So what does this additive regions mean? For example, now you have to see that these particular, when the double digestion is occurring, then it is yielding particular regions, right? The sum of these regions should be equal to regions that are obtained individually by single digestion, right? For example, if you say eco R1, right? Let me just write it down somewhere. If I say eco R1, right, it yields a 3 kb fragment. Hold on. And a 3.5 kb fragment, right? Over here, if in this restriction digestion, this 3 kb fragment could come from this 2 kb plus 1 kb, right? So 2 plus 1 kb fragment could give this 3 kb fragment, and 3.5 could come from the individual 3.5 kb fragment, right? Now the other additive region for Hind 2, what will it be? Hind 2 has basically a 2 kb fragment and a 4.5 kb fragment. So this individual fragment could yield a 2 kb fragment, and 1 plus 3.5 can yield a 4.5 kb fragment, right? So this is how we need to look at the question, right? So we have found the additive regions over here. Additive regions have been found between the single and double digestions. Now we have to find the overlapping regions, right? So if this is going to be a linear vector, right, we have to see that in eco R1, there was this 2 plus 1 kb fragment, and in this Hind 2, there was this 2 plus 1 sorry, 2, 1 and 3.5 kb fragment, right? So, okay, where do I write this? This 2 plus 1 is coming over here for equar 1. Then this 2 is coming separately. Then this 1 plus 3.5 is coming for this particular thing, and this 3.5 is coming separately, right? So it will probably be something like this, that a 2 kb fragment is present, then 1 kb fragment is present, then 3.5 kb fragment is present. You understood how I did this? I basically saw what is overlapping, right? There is 1, 2 plus 1 kb fragment. Then there is 2, so I will put it below 2, right? Then there is 1 plus 3.5, I will put it below 1, right? 1 below 1 and then 3.5 separately. And there is one only 3.5 kb that I will put under 3.5. So this 2 kb will come on one end. This 1 will also overlap, and this 3.5 is also overlapping, right? So this is probably what we will basically get, right? I hope this particular one was also clear. Now, how will we mark this? We need to mark this also, no? So this 2 kb we are getting from Hind 2 and 4.5 kb, right? So Hind 2 will probably break over here. It will give 1 2 kb and 1 4.5 kb, and eco R1 will probably break over here, which will give one 3.5 kb and one 3 kb fragment, right? So this is how a restriction map for this particular question will be drawn. I hope it is clear. Okay, so this one is a final question. We have sufficient time to solve it. Okay, so over here, this is basically a representation of a gel electrophoresis result, right? After the DNA has basically migrated, this is the representation. Sorry, this is the representation. Let me just get the pointer. This is the representation, right? So now this is the key for it, right? So it says that lane 1 is ladder. Lane 2 is the undigested one, right? Lane 3 is eco R5 digestion. Lane 4 is BAM H1 digestion. And lane 5 is double digestion by eco R5 and BAM H1, right? Now, what do we have to do? Using this particular information, we have to draw a restriction enzyme site map of a plasmid using the given data, right? So, and over here, upon these bands, they have mentioned the fragment size. Okay, so these are different fragments that have been obtained. First, let me just write it down. Which one is which, right? For our ease later. So, lane 1 is basically ladder. Lane 2 is undigested. Undigested means that the complete fragment is going to be this huge, that is 700 kb, right? Then third is eco R5. Okay, eco R5. This is BAM H1. And okay. Oh God, what did I do? This is eco R5 plus BAM H1, right? and the size of these fragments are mentioned on top of the fragment, right? So we know that the undigested fragment is 700 kb, so the entire thing will be of 700 kb, right? Now we can individually draw the this digestion sites for EcoR5 and BAM-H1, right? If we individually draw it, it will help us for drawing the final restriction enzyme site map. So I have drawn it for your convenience. I've already drawn it. So EcoR5 has two fragments. One is 200 kb and one is 500 kb, right? So I have already drawn it. One fragment is 200 kb, the other is 500 kb, right? Similarly, for BAM H1, one fragment is 250 kb and the other is 450 kb. So for BAM H1, one fragment that I've drawn is of 250 kb and the other one is of 450 kb, right? Now now for this double digestion, we need to see how will we draw this particular final plasmid, right? With restriction enzyme sites. So over here, we need to see that are there any restriction sites that map with any particular restriction sites of EcoR5 or BAM-H1. So these two restriction sites are the same as that which is present in BAM-H1 and 200 and EcoR5. That there is one particular segment Okay, there are a lot of technical glitches today. Okay, I'll try and complete my question as soon as possible. Okay, I've presented my screen. I hope it is visible to everybody. I'll go on the presenter mode again. Okay. So I am taking that it is visible to everybody and I'm going to redo this question, right? So this question basically said that using this data, I hope I don't have to discuss the entire thing again. So using this data that there is this electrophoresis result, right? And there are different,
Hello.
yeah.
Yeah, you're not visible, Arveen.
Oh, I'm not visible. Okay.
Even the presentation has been paused 3 minutes back.
Okay. Is it visible now?
No,
No, the presentation is not visible.
no.
Okay. Is it visible now?
No.
Yeah, I'm not sure why this I'm running into this problem. Just let me know whenever it gets visible.
Yeah, I'll let you know.
Okay. Now is it visible?
No, no, no. Everything is dark.
Oh, okay. I will probably stop sharing and I'll rejoin. Maybe when I rejoin, that was the issue. Hold on. Is it visible now?
No, Vin.
Oh God. Okay. I'll maybe try once or twice. Otherwise, what I can do is I can just include, I have included the answer for this particular question also in the presentation. Maybe then you can just read the presentation. I'll not be able to actually explain it, but I'll try. Okay.
Yeah, not a problem. You can upload it at your own time of choosing.
Can you see if I am uploading it like this? Wait. Now is it visible?
Yeah, yeah,
It's visible. Okay.
yeah.
I like this. I don't need to share my screen, but this is visible, right?
Yes, as of now it's visible.
Okay. All right. So I will try to explain it without actually writing it down, if that is possible. I don't know. Okay. So I hope the question was visible. I will go through the explanation once again. So over here, what is happening is that there is this particular gel electrophoresis result, and in lane 1 it basically signifies the ladder, right, which is basically going to tell us what molecular weight fragments these are, right. The second lane is going to be the undigested fragment, which means that our entire plasmid is of 700 kb, right, because it is undigested, it has not been fragmented, so we know that the entire basically uh, kilobases of this particular plasmid is going to be 700. Now the third one over here is 500 and 200, which is Eco R5 digest. So if this particular plasmid is digested using Eco R5, then we receive two fragments. One is of 500 kilobases and the other is of 200 kilobases. Right? If it is digested using BAM H1 in the fourth lane, then we again obtain two fragments. One is of 450 kilobases, the other is of 250 kilobases. Right? And if there is a double digest using Eco R5 and BAM H1, we obtain four fragments. One of 250 kilobases, one of 200 kilobases, the other two are of 150 and 100 kilobases respectively. Right. So to solve this, I first said that there are two things we have to keep in mind. One, the entire plasmid, which is undigested, is of 700 kb, right? Which we can see over here also. 500 plus 200 will give 700, 450 plus 250 will give 700, and the addition of these four fragments will also give 700, right? The other thing is that if we individually draw Eco R5 digest and BAM H1 digest, it might help us, right? So that is what we have done in our next slide. So over here in Eco R5, there are two fragments obtained. One is 200 kilobases and the other is 500 kilobases. After this, if we draw BAM H1, again two fragments are obtained. One is 250 kilobases and the other is 450 kilobases. Right? Now, going back to the question, when we see this double digest, right? In the double digest, we have to see that are there any fragments that coincide with the fragments of the single digest, right? So yes, there are two such fragments. One is 250 kilobase fragment, which coincides with 250 kilobase fragments of BAMH1 digestion. The other is 200 kilobase fragment, which basically coincides with 200 kilobase fragment of this Eco R5 digestion, right? So this basically means that out of the 700, these two fragments are two different entities, right? So this Eco R1, R5 digest and BAM H1 digest will take place at two individual levels, right? So uh, after, okay, I wanted to draw it step by step, but I don't know if I use this. Okay. If this is basically plasmid, so what it means is that there will be one 200 kilobase per fragment, right? 200, and there will be another 250 kilobase per fragment, which is definitely going to be present, right? This 200 base pair fragment will come from what? It will come from Eco R5 digest, right? Eco R5. I'm just going to write Eco over here. Okay. And this one is going to come from BAM H1, right? So this we have sorted out. After this, there are two fragments that are left, 100 and 150, right? So we can place it anywhere because it is not really going to affect the restriction sites now. So we can say that this one is a 100 kilobase per fragment and this one is a 150 kilobase per fragment, right? So the total is going to be how much? 700. How? 150 and 250 will be 400 and this 200 and 100 will be 300, which will contribute to 700 kilobases. This entire plasmid will be 700 kilobases. It will contain two Eco R5 sites and two BAM H1 sites, which will basically divide the plasmid into four fragments, right? When the double digestion is done. When the single digestion is done, then this Eco R5 sites will divide it into two fragments. One will be of 200, the other will be of 150 plus 250, 400 plus 100, 500, right? 500 base pairs. And the in BAM H1, one fragment will be of 250 kilobases, the other will be of 150 and 200, 350, 350 and 100, 450, 450 kilobases, right? So that is what our answer is also going to be, right? Now it is not necessary to write 100 over here, 150 over here. It could be the other way around. It will not really be a problem. You can write it in that way and you can find it out, right? So I hope this particular problem was also is also clear to people who have stayed back. So hopefully it is clear. If not, then please let me know. Otherwise, this was my final problem for today, and that is it for today's lecture, right? So yeah, maybe if you guys did not have a problem, you might leave. I'll just save the recording for the lecture, and then I'll also leave. All right. Thank you, guys.
Thanks.
Thank you.